Hello everyone, welcome back to Dr. K. Prem Primer Lecture Series presenting by Dr. K. Prem, that's me. In today's episode, I'm going to talk about alpha complementation. It is also called as a blue-white screening. Let me define the what is alpha complementation. It's a alpha reconstitution of functional tetrameric beta galactosidase with alpha peptide from vector and omega peptide from the host E. coli is called as alpha complementation. It's reconstitution of tetrameric functional beta galactosidase with alpha peptide from the vector and omega peptide from the host E. coli is called as alpha complementation. To understand the concept of alpha complementation, we should be aware of three things. They are beta galactosidase and the vector and E. coli. The first thing you should be aware of what is the beta galactosidase and its properties. And the vector, here vector is pUC18 and the E. coli host. If you can understand these things, these three things, and it will be peanuts to understand the alpha complementation. So first, I'll explain the beta galactosidase. Beta galactosidase is an enzyme which degrades the or hydrolyzes the lactose into galactose and glucose. Right? So when you look at the structure of beta galactosidase, it is having two domains, alpha domain at the N terminus, omega domain at the C terminus. Alpha domain extends from 1 to 140 amino acid and the rest is omega domain. So whenever you degrade from 1 to 140 and that peptide, if you mix the alpha peptide and omega peptide again, it will form a functional beta galactosidase. So here, if you cut in alpha peptide, if you cut first 10 amino acids, then rest is, if you leave it, then if you mix it again, first 10 amino acid can mix with the rest of the beta galactosidase to form a functional beta galactosidase. So this is a beautiful property of beta galactosidase, which is helps in the designing of alpha complementation. And you see here alpha peptide is degraded and omega peptide is there. When you mix both of them, and individually they are inactive. If you mix them, if you divide them or degrade them chemically or proteolytically, and it forms two parts, alpha and omega, they're independently inactive. But if you mix them again, they form a active tetrameric beta galactosidase. Right. So here the alpha part is encoded by the LAC-Z prime and omega peptide is encoded by the rest of the LAC-Z. So LAC-Z prime is incorporated to the vector and mutated rest omega peptide encoding gene, that's a delta LAC-Z is there in the host, that's E. coli. The same. This is all about the beta galactosidase. Next, we will see the vector. Here, we use the pUC18. That size is 2686 base pairs, and it is having four important properties, as you all know that. The first one is the PLAC is a lactose promoter, and here under the promoter, there is a LAC-Z prime region which encodes the alpha peptide whose length is 30 amino acid. So in the LAC-Z prime region, there is a MCCS, a series of restriction sites which facilitates the cloning of various DNA fragments. So this alpha laxad prime region encodes the alpha peptide from of beta galactosidase. And the second property is ampicillin resistance marker, which provides the resistance to the host in the presence of, it can grow in the presence of ampicillin. And origin of replication is required for the autonomous replication of plasmid in a host, irrespective of the genome replication. This is the second important element to understand. And third one is a laxed M15 E. coli strain, that's a mutated one. In its genome is mutated where uh, 11 to 41 amino acid encoding fragment is removed, and the rest of the fragment is there. So it is lacking the only 30 amino acid of beta gal, and rest is there. So that 30 is there in the pUC18. So whenever your pUC18 is transformed into the E. coli strain with the mutated laxed region, then what will happen? We see in the next slide in the presence of IPTG and and XGOL. So this region is mutated laxed and encodes the omega peptide of a beta galactosidase, which is almost 993 amino acid. So this is the laxad mutated M15 equal strain, and I'm going to explain the concept here. When this strain is transformed to E. coli, and when you grow them in the presence of beta, in the presence of XGAL and IPTH, what will happen? The first we will see the what in the absence of IPTG. In the absence of IPTG, RNA polymerase binds to the repressor promoter and transcribes the repressor gene. And that mRNA converted to the protein. That protein is a repressor protein. That repressor protein is very active, and it's also tetramer. It can bind to the operator region of promoter of the laxed gene. Laxed gene. This is a mutated one. This is a mutated one where 11 to 30, 41 amino acid is mutated, rest is there. And this active repressor goes and binds to the operator of the laxed promoter. Laxed promoter. And once the repressor binds to the operator region, and it cannot be identified and bind by the RNA polymerase, then there is no transcription of laxed gene. So this is all happens in the absence of IPTG or allolactose. Whenever you add IPTG or allolactose, what will happen is IPTG molecule can bind with the repressor. You see, it is bind with the repressor, and it will inactivate the repressor. So bound IPTG, this complex repressor plus IPTG molecule is inactive. It cannot bind with the operator. Instead, it dislodges from the operator. You see, right? Now the operator region of laxed promoter is free. In that condition, RNA polymerase comes and binds to the promoter and transcribes the laxed region, laxed gene, and mRNA is synthesized. The laxed mRNA is synthesized, and it encodes it translated and produces a protein that's an inactive omega peptide beta galactosidase. So as you all know that the E. coli strain is having the only omega peptide encoding laxed region. The alpha region is present in the vector, right? Now the vector is also vector promoter also recognized by the E. coli RNA polymerase and transcribes the laxed and synthesize the alpha peptide. This alpha peptide associated with the omega peptide of beta galactosidase and form a functional tetrameric beta galactosidase, right? So to know when you transform the PUC18 into the E. coli host, which is having the omega peptide, and vector is having the uh, alpha peptide encoding gene, when they are when they form the respective peptides and they associate, and to understand, to know the system is having the active beta galactosidase, there should be indicator. Here they are provided with the X-gal, right? It's a 5-bromo 4-chloro 3-indolyl galactopyranoside. This is one of the substrate of beta galactosidase. When beta galactosidase acts on the X-gal, it breaks down the glycosidic bond to form a galactose and indole. Galactose and indole. So the indole immediately dimerize and form a insoluble blue color, forms the insoluble blue color. So whenever a E. coli strain which is transformed with the pUC18 with the R plated on a IPTG plus X-gal, the colonies looks blue in color because the active enzyme produced with the alpha of vector and its genome, and the functional tetrameric beta galactosidase hydrolyzes the X-gal to form a blue color. So the colonies looks blue in color. This is the basis of alpha complementation. I'm saying reconstitution of functional tetrameric beta galactosidase with the alpha peptide from, this is a functional tetrameric beta galactosidase form with the help of alpha peptide from the vector and omega peptide from the laxed genome, and form a active beta galactosidase. This is called as alpha complementation. Hope you understand. So this became a screening method for selection of recombinants from non-recombinants. I'll show you that in the next slide. Right here you see this is a E. coli genome with the omega peptide. If it is transformed with the self-ligated vector, means self-ligated vector means only vector recircularization, so no insert. You see, this is the self-ligated vector where laxed prime is intact, there is no insert within this one. What will happen? Laxed is transcribed and produces the alpha peptide. So alpha peptide and omega peptide both associates and form a functional tetrameric beta galactosidase. So E. coli with the omega peptide and vector with the alpha peptide can form a blue colonies on the media with the IPTG and X-gal. You can see blue colonies, right? But in other scenario where E. coli, which with the omega peptide is transformed with the recombinant plasmid, means a gene of interest is cloned into the MCCS of multiple cloning site of laxed prime region, then what will happen? You see? You see? Here, pUC18, a laxed region where in the laxed prime region, a gene of interest is cloned with the help of MCCS, right? So that what will happen? when it lacks that region is transcribed and it cannot encode a normal alpha peptide, then the alpha peptide is may be formed, may not be formed. When it formed also, it loses its conformation. It loses its conformation. The conformational changed, conformation structurally changed alpha peptide which found because of insertion of a gene into the laxed prime region, then that functional, the conformation changed alpha peptide cannot bind with the omega peptide. So the alpha peptide structure has changed because of insertion of DNA into the MCCS of laxed prime. So there is no functional beta galactosidase when E. coli is transformed with the uh, vector with the insert, right? So there is no functional beta galactosidase from this E. coli. So these kind of E. coli are plated on a LV media with the IPTG and XGAL. They looks white in color because of not having the functional beta galactosidase. So the blue colonies are non-recombinants. The white colonies are recombinants. So recombinants don't have functional alpha peptide because of insertion of a gene. Then it cannot associate with the omega peptide. There is no formation of functional beta galactosidase, and because of lack of the beta galactosidase, it cannot hydrolyze the hexagal to form a galactose and indole. Then there is no blue colonies. So only non-recombinants can have the functional beta galactosidase and produces the blue colonies in the presence of IPTG and XCAR. But recombinants cannot have a, there is no possibility to form a functional beta galactosidase because of insertion of gene. Then these colonies appears white in color. So this is how you can distinguish the recombinants from non-recombinants. Single step method, simple and visually distinguish between the colonies of blue color and white colonies. So there is no extra step to required for the selection of recombinants and non-recombinants. If you go by chronological order in a text format, you can see beta galactosidase is a tetramer. Each molar contains a 1023 amino acids. Each monomer have alpha and omega domains. Laxed prime gene fragments encodes a alpha peptide inserted into the vector pUC18, and remaining 990 amino acid encoding mutated gene is present in the E. coli strain. When pUC18 is transformed with E. coli and plated on LV media with IPTG and XGAL, IPTG binds to represent, allows the RNA polymerase to bind to LAC promoter and transcribes both LAC-Z prime and LAC-Z regions from the respective system. LAC-Z prime transcript translated into the alpha peptide, then alpha peptide associated with the omega peptide, which is derived from the E. coli host, forms a functional beta galactosidase. A reconstitution of functional tetrameric beta galactosidase with alpha peptide from vector and omega peptide from the E. coli is called as alpha complementation. That galactosidase acts on X-gall and hydrolyzes it form a blue colonies. When we clone our gene of interest into the MCCS present in LAC-Z, due to insertion, alpha peptide loses the conformation and cannot associate with the omega peptide. That means no functional tetrameric galactosidase form. Hence, the bacterial colonies looks white in color and media with the X-gall and IPT. So this is all about alpha complementation. Hope you like this lecture. If at all, please try to subscribe my channel. That's a Dr. K Primer. And if you wanna, you can share. And if you like it, give thumbs up. And if you have any issues, please let me know through the comment section. Okay, this is all about alpha complementation, and try to subscribe so that you will not miss any update from my end. Thank you all for listening. See you again. Until then, bye bye.